I could not contain myself to the end because this is just absolutely crazy. And this is the game that these guys play. And then the silly YouTubers look at this and they say, oh, these guys got it because they claimed it in their paper. And this is what I say. When you look at the experimental details, it actually argues against it. So when you publish papers, if you don't come clean with this, then what happens when you don't come clean is the layperson, like the YouTuber, picks this up and is totally misinformed on this because it's actually just the opposite. This is what I'm talking about. This just nails it home. And again, plus there's a ton of user input. Plus this couldn't work because they've got the selective activation. There's all these games that they're playing. Okay, well, I'm here today with Dr. Royal Truman, and he is a PhD organic chemist, and I'm going to let him introduce himself and tell us a little bit about his background. But let me say that we're going to be talking about this amyloid hypothesis today. There's been some talk about how amyloid hypothesis solves the origin of life issue, and so we've got an expert here that can talk about it. And so he's got a PhD, and he can address this. And let me first introduce to you Dr. Royal Truman. And Royal, good to have you with us here today.
Yes, Dr. Torrey, thank you very much. It's actually a very big honor for me to be here.
And could you tell us something about your background? Where did you get your degrees, and what have you been doing for your career in regard to organic chemistry?
Okay, my formal degrees are bachelor degrees in both chemistry and computer science from SUNY Buffalo. Got a doctorate from Michigan State. Also an MBA from University of Michigan. After beginning work for BASF, which is the world's largest chemical firm, I continued working with various universities, continued studying bioinformatics and other things. So have had quite a rich experience working for BASF. I grew up in several countries, speak several languages. So at BASF, I was also able to work in different countries. My research has been in many areas and background, not just organic chemistry. I'm passionate about molecular biology. I would say one could call me a data scientist. That means that my specialty has been understanding data and all kinds of methods to analyze it.
Okay, good. So you certainly sound qualified. So what I want to do is I want to release to you to just go ahead and give a seminar. Now let me just put this up front. We don't know how to really dumb this thing down too much. We're going to have to talk about chemistry. We're going to have to talk about how molecules function and how they work. And so I'm going to let Dr. Truman go on this. I'm going to try to not interrupt him very much because he's got a lot of data to go through. And then after he's done, we'll discuss it, and we'll discuss the ramifications of this. But I want to say that this involves chemistry. It involves details of chemistry. If you've not seen my 14-part series, you can see that on Origin of Life and learn the chemistry where I really tried to explain it. This is not going to be that. This is not where we're going to stop every minute and explain what this means. We're going to go through and we're going to explain this amyloid hypothesis and explain how these things work and what are the problems with it, and then we'll discuss it afterward. But this is going to be a robust seminar on chemistry. And this is the only way, this is the language of how cells operate. And so that's what we're going to go through. So with that, Dr. Truman, I turn it over to you.
Okay, thank you very much. Yes, I will try to compromise a little bit, meaning I have to give enough details to be of some value. But I don't have to overcomplicate things. So that said, I am including a lot of text, diagrams, molecules, structures in the slides. So of course, people can go back and refresh their memories and think about it. I want to mention also that I've written over a dozen papers on this topic, and all of them are referenced at the very end of the presentation, also with URLs. So the more technical, more challenging things are actually in the papers for those of you who are, let's say, more advanced in this material. So this won't be too daunting. I decided to split it in two parts. The first will be just the topic, are amyloids relevant to explain the origin of life? And then the second part is addressing the question, would amyloids realistically have existed abiotically? So let's jump into the first part. Are amyloids relevant to explain the origin of life? So here's the structure. The first part, what are amyloids and what is the Amyloid World Hypothesis all about? Why might they be relevant? What are the properties? And I'm going to talk about some key studies that are often referred to from which the major conclusions are drawn. These studies will go into, especially the first one, a little bit of depth. And then I'll wrap up part one with some key observations that are relevant to all such amyloid experiments. What are amyloids? Well, they're protein aggregates that are produced when beta sheet structures are stacked together. And stacked sideways, they form these long fibrils, these long strings. Okay? So these protein strands or part of the proteins are stacked sideways. And when this happens, it causes nearby proteins sometimes to also misfold. That's a general idea. Now, some key names associated with the Amyloid World Hypothesis. A Professor Rieck, who is a chemistry professor at the ETH Zurich, a very famous university, a very well-known, very distinguished professor. Also, a Professor Maury, who worked for many years in medical research at the University of Helsinki, and Professor Gasset at the University of Tel Aviv has done a lot of theoretical, conceptual work on amyloids, perhaps not so much the amyloid world hypothesis per se, but it's provided some of the background knowledge. So why are amyloids of any interest in this whole topic of origin of life? Well, we have a big problem, and that is RNA, DNA, and polypeptides hydrolyze in water. That means it's pretty hard to envision any kind of scenario where life could have originated on its own in water, given the chemical realities. Amyloids would be stabler if they could form. Once they form, they would at least persist a while. Another problem with origin of life chemistry is that one needs to have polypeptides or protein structures that fold properly and reliably to have any kind of a biological-like function. That is a very, very, very big problem because the vast majority of protein sequences don't do that. However, amyloids can affect the structure of nearby polypeptides. So thinking is, well, if you had amyloids, they could then force nearby peptides to adapt to their shape, and that would perhaps alleviate that particular issue. So how does it all fit into the various thinking? Those who believe in the amyloid world hypothesis suggest, fine, let's stick it here somewhere in the middle before an RNA world, which again is hypothetical. And that would have preceded our DNA world and the last universal common ancestor. And before that, then you'd have various chemical processes going on. So all of this would have occurred a long, long time ago, like 4 billion years ago. That's the thinking. So only parts of proteins are typically involved in causing amyloids. Amyloids are well known to cause various diseases. So think of them more or less as abnormal misfolded proteins. Alzheimer's disease and other diseases are caused by that. Now, for the amyloid world hypothesis to be plausible, the peptide should be as short as possible. Otherwise, it's simply not credible at all that these amyloids could have formed. It is true that very few peptide sequences will actually form the requisite beta sheets, which when combined in large numbers can lead to amyloids. However, biochemists understand very well how to engineer peptides to form beta sheets. I won't go into that, but I do mention some of the tricks here. There, where you put what kinds of amino acids will allow you to form beta sheets. Beta sheets, alpha coils, these are substructures out of which proteins are formed, and they're necessary before proteins can fold properly. So a lot of very clever people have experimented extensively trying to find the shortest peptides possible and have succeeded in reporting peptides as short as 4 to 7 amino acids, AA amino acids, that can form amyloid-like substances if in very, very high concentrations under special laboratory conditions. So once again, the trick here is one has to cleverly find sequences that will form beta sheets, and they should be as short as possible.
If you do not believe in the physical resurrection of Jesus Christ, send me an email, tour@drjamestour.org, and we will get together, and I will share with you about why I embrace the resurrection of Jesus.
Now, why might amyloids be relevant for origin of life purposes? I'm going to talk about some key studies, one in particular by Root et al. We'll spend a little time on that because it is referenced a great deal, and a lot of the general conclusions stated about amyloids is based on that one study. So we read in a key paper here that amyloids composed of short peptides can direct the sequence-selective, regioselective, and stereoselective condensation of amino acids. What is being said here? Let's look over here at the structure of an amino acid, okay? You've got a carboxyl group on one end, amino group on the other end. We have several issues here. One is, of course, the chiral center here. So you've got L and D enantiomers, and you want only L to be biologically relevant. That's one issue. Another problem you've got is that different parts of the molecule can react. For example, you've got a side chain here, and depending on the amino acid, sometimes you've got a carboxyl group down here in addition to the end position, and the same is true of an amino group at the side chain in addition to the end position we're supposed to react. So what are these people claiming? First of all, sequence selective. What are they saying? Sequence selective. If you condense a bunch of amino acids together, they're going to react more or less randomly. That means there's very selectivity as to which amino acid would condense at either end of another amino acid. So if you end up with a mishmash of random amino acids, it's pretty hard to foresee how you could get anything useful. So they're claiming to have done some experiments that perhaps address the issue of sequence selection, that in some manner amyloids are able to preferentially select or condense some amino acids and not all randomly. So that's the first claim. Regioselectivity is referring to the need to form long linear peptides, which means only the N carboxyl or amino groups react, but not the side chain. And the stereoselective issue is referring to L or D amino acids reacting. So, as I mentioned before, for all the things that we were talking about today, I, with other people, have written papers. So the references are used in the slides, and at the very end of the presentation, I'm also giving the URL so you can download and look at a much more detailed analysis of all these topics. So here is the key experiment by Root et al. Here's what they did now. They very cleverly, I mean, they are clever people, put together two kinds of peptides. One is called a template. It's an 8 amino acid template combined with substrates that were between 5 and 7 amino acids long. Here's what they did. Here is the template peptide, and it was mixed together with a substrate. So here's one substrate, here's another substrate, here's another one. So we'll call this experiment series number 1, number 2, number 3. What is important and significant about this design? It is the fact that the template is 1 amino acid longer in this case than the substrate. And here it's 2 shorter and this is 3 shorter. So there's an overhang here. So you've got two kinds of peptides. You mix them together, and they are very, very carefully designed so that they form beta sheets because of the pattern of hydrogen bonds and hydrophobicity and hydrophilicity. So the particular pattern of these two components allow them to mix together, and at very high concentrations, they form sort of like a crystallization, they form amyloids. So that's the key. So what we must not overlook is the design feature of this particular overhang here that distinguish the two parts of the peptides. Unfortunately, I have to mention something that is true of all the experiments I'll be talking about and all the experiments you'll probably ever encounter in this field. The peptides were chemically modified. These are not peptides you would find as is in water form naturally. That's why you see that the carboxyl and the amino ends here look a little funny. It's because they've been protected to keep them from hydrolyzing or degrading in any manner. And that's also true of one end of the substrates, and the other end is left open so that an amino acid can then react at the end here. Question?
Yeah, this is interesting. So even in this experiment where they're citing, this was a loaded system, is what you're saying. The one end was it protected or activated? I mean, this seems very strange.
It's always done that way, and the answer is yes. It's both activated and blocked. Oh. The amino acids here that are going to react here or here or here, those amino acids are activated. CDI, they're activated because these things, for reasons I'll go into in part 2, are thermodynamically unfavorable. These are in water. This is not what would happen. So you got to activate them.
Yeah, I want to let you go on. It's just, this is such a cheat, and to somehow suggest that this is relevant to origin of life when you're doing this selective blocking and activating. It's. I'll just call it a cheat. We'll talk about it more at the end. But go ahead.
Yes, it's a good observation. That is correct.
Yeah.
So, to summarize, both ends are blocked in the case of the template. One end in the case of substrate, and the other end is left open to allow it to react. But then the amino acids that are then added to the once formed amyloid are then in a high energy state to allow them to react. So, to repeat a little bit what I said, perhaps help you visualize it. Think of these two peptide components that are very carefully formed. For example, the phenylaniline, the F here, are not aligned across from each other. Otherwise, you could never form the amyloid, but they're shifted by one amino acid. So that's why I say it's complementary. It's designed so that the two peptides together will form an amyloid. And then think of this like as a puzzle where a piece is missing. Okay? So now the trick is, can chemists get some amino acids to react here preferentially to others? That is the whole theory, the whole idea between sequence selection. They're trying to find some mechanism, some way to show how the sequence of peptides is not totally random, as you would pretty much expect, but there could be some being favored over another. And the answer is, well, yes, chemists are, clever people and they can design things to do interesting things. If you have the right amino acid here, the right kind, meaning the side chain complements what would be at the opposite end here. If it's the right size, etc., it would elongate, it would extend the beta chain by one position. That would be thermodynamically attractive, and it would be possible. So that was the theory. So what they did then is added 5 different amino acids with different properties, and these were D and L mixtures. So D and L mixture of phenylaniline, aspartic acid, glycine only comes in one flavor, valine, arginine. Okay? So, and here are the results. To refresh your memory, there is one position open, and it is sterilely, in 3-dimension, prepared in such a manner that in the amyloid, an amino acid would fit very, very well there. And lo and behold, as expected, the phenylaniline does fit in and does react there preferentially compared to other amino acids. And here is the significance. They compared the amount, the yield of phenylaniline that would add to the substrate alone, which is the first value in each pair of the table, that yield, and compared it to how much of the same amino acid would add if the amyloid had first been formed. So, if the phenylaniline is added to the substrate alone, the yield is very low, as expected, but it's quite high in the case of the amyloid. So, that drew a lot of attention. Similar but much smaller effect in the case of valine, which is also hydrophobic, and therefore it would also complement that particular position, and it was not true of the other amino acids.
So just to clarify, what are the favorable yields that they're quoting here? It's gone up from what to what? Went without the template to the template. How much did the yield increase?
Basically, the ratio here is what you want to look at.
The ratio.
Yeah. So there's no question that in this particular amino acid, that that the amount that they were able to obtain of the addition product versus nothing happening. You just wait for a long period of time until the reaction is no longer adding anything, and once it's plateaued out, you say, okay,
Yeah.
under these conditions.
So the ratio is that
Yeah.
is that 2.8 to 50 or something?
Exactly.
All right. So you have 2.8 has gone up to 50. That ratio is 50. So in other words, it's. Do they have any idea of the yield here? What does 50 mean?
Trying to remember exactly
The reason I say this is that
Right.
Years ago we had a project where we were trying to make a mother-daughter duplex, something very similar to this, but not using biological entities, and the fidelity killed us. We would have 30% of the non-correct one, and then that non-correct one became a template as a new mother for making other things. And within a couple of cycles, the whole thing is absolutely unusable. These guys are using biological entities. So you'd think that, they're taking advantage of biology, what biology has worked out here. And unless they're going to tell me what the yield is and what the selectivity is, they're just showing the ratio. It was 2.8 before, it became 50 now. So
Right.
it was higher, but if it's not at 99.9% of the one they want, fidelity's going to kill them. But if they're at 50% of what they want, this is a bunch of nonsense, utter nonsense. So tell me where they are on this thing.
Right. I'm trying to remember exactly how the yield was mentioned. My recollection was it was the amount of activated amino acid that was available and then how much had been consumed. So it is indeed a high yield, but that is not really the fundamental problem. You're right that there are some serious problems with it, several problems, but and in fact you're alluding to it correctly. It's a yield problem, but the problem is actually a little bit different than you suspect. In fact, let me jump the gun and just kind of give away a bit of a spoiler here. And you'll see a little more quickly what a much more significant problem is. That's the following. And it's something I'll come back to and quantify and look into more depth. But here is a very fundamental issue. You have here two peptides that are in extraordinarily low concentration in nature. Low enough, so I'm telling you right now, you'll never find it. But forget that. Just say very, very, very low concentration. Now, here's the issue. These two things were first mixed together to form the amyloid before the amino acid was added. Now, let's talk about the real world. In the real world, you've got this peptide somewhere in South Australia, and you've got a couple of these guys over somewhere, near Chile. And then you have, don't ask me how, some activated amino acid. The amino acid is going to react almost always immediately whenever it can, and not wait until this plus the amino acid are all together at the same location. That means
they've conveniently put everything in the same reaction tube to react, and
exactly.
The things are not like this. But we'll give them that, even that. But now you have these conveniently activated. This is huge human input that would not be available on Origin of Life. This is a bunch of nonsense when it's applied to Origin of Life. And I'd love to see the yields here to talk about the fidelity of this, because if it is not very high purity, this whole idea of templating and reproduction is toast. It's over. But anyway, press
I'm
on.
with you, but let
Press on.
me be fair
Yes.
to them to at least make sure that we understand what is being argued, and then afterwards go into the plausibility aspect, because there's several issues here. But it's important that we first understand what they're trying to do. They're trying to say, if everything was set up the way it was here, we have an example where a particular amino acid here would preferentially add, and other things that were not hydrophobic, other amino acids would not as readily. Okay, let's just say that's their thinking. First, we'll understand their thinking, and then we'll see if there's any merit to it. In very similar experiments, two of the amino acids are shown here, and now you've got three other ones that were not shown before. Now, they are not looking at the relative yield, but the preference for the L amino acid when reacting with the substrate alone versus the preference for L in the case of the amyloid. So, amino acids come, because they're racemic in two flavors, except for glycine. So you would expect them to react 50-50. We've seen here, a little surprisingly perhaps, that 0.6, meaning the ratio is not 1, but 0.6 is of the L form is added to the substrate in these 5 examples, but a lot more of the amyloid. What is that saying? Let's go back for a second. All that is saying is that in order to permit the beta sheet to be elongated, you need the L form. And that's why, indeed, the L form was preferred in the case of the amyloid, but not in the case of the substrate alone.
Yeah, but what's the ratio of the preference of the L over the D?
You see,
It's very hard for me to see that. It's really small text. It's hard for me. The screen is far from me. What is the ratio?
Okay. So in the case of substrate alone, it's less than 1, but it's significantly over 2 in the case of the amyloid.
So the ratio is 2 to 1.
At least, usually more than that.
I mean, if you only have a selectivity of 2 to 1 in this kinetic separation between these, this would be laughed at. I mean, it's laughable because you're doing this multiple times. So you keep inserting 30%, 33% of the wrong isomer. Every time you've inserted a wrong isomer, boom, it's done. It's over. So this is not a templating effect. I mean, it's so poorly done that this is not a daughter from that mother. This is another molecule. So this whole idea that you're copying, you're not copying at all. If these guys don't have the pure amino acid, the enantiopure amino acid to start with, right, it's toast again. So this is another reason why this is nonsense. Go ahead, press on. I'm just trying to just give a little short critique here, but I don't want to slow you down too much. Press on.
That's true.
Because of you, millions of people have heard the gospel. If you confess with your mouth Jesus is Lord and believe in your heart that he's risen from the dead, and you will be saved. That's right. That's the requirement. We talk about science concepts which draw people in. Take these nanomachines and have them drill into cells. This would be a great way to kill cancer, right? We also talk about Jesus Christ, who's the best in everything. My faith in Jesus Christ means more to me than anything. If you could continue to give or give for the first time, we would certainly appreciate it. You can go to jesusandscience.org. Thank you so much.
Okay, I do have another spoiler here. And again, the question is, what would actually have occurred prebiotically? What the authors did not point out, I will have to, what I find very remarkable, and I hope the font is large enough so that you can see this, in every pair of reaction, when comparing the substrate versus what happened in the amyloid, the first of each pair is a substrate alone. So L to D, 0.6, 0.98, 0.9, 0.7. Think about this. This is very interesting. You did not have a 50-50 mixture being added to the substrate. You actually have less L being added than you would expect by chance. Remember, you want L only? So what is going on here? How come we're getting less L than you want by chance being added to these substrates? And is that significant? And that is enormously significant, even though it wasn't mentioned in the paper, but the data is showing this. It's because it's a recurring phenomena I have noticed in these kinds of origin of life paper. And that is, you just can't outwit entropy. Here's the problem. These substrates are L only, right? So here's a 7 amino acid L only. And when you got all of these L only, it forms a structure that actually decreases the ability of another L from adding to that particular substrate. It decreases it. So that means that in the real world, where the substrates reactions would dominate substrate plus amyloid, because substrate plus amyloid is bimolecular, and the chance of two very, very low concentration molecules being in the right place and time together is, of course, vastly, vastly lower. Therefore, in the real world, the dominant outcome would have been reaction of the substrates. And the substrates are now saying, guess what? If you got really, really, really lucky and some of your peptides had L-only amino acids, you end up decreasing the chance of adding another L to it.
Okay, so let me just cut to the quick here and just give the explanation of this. This is tremendous. How anybody can look at this and think that this is relevant to origin of life is just, to me, absolutely idiotic. Here, what he's showing then is that if you have the all-L, which is what you're going to need for the natural system, it disfavors being a template for another all-L configuration.
Exactly.
It actually is favoring the opposite antipode, the opposite enantiomer from coming in. That's what's favored. So again, this argues against this being relevant to the origin of life. This very data, their very data argues against it. This is what I say over and over again. I look at the same data and I say, that tells me that's not how it happened. This is not how it happened. So anybody who would look at this and study this and look at this, as you have said, you'd have to say this argues against the origin of life. Anyway, I digress. No, I don't digress.
No, that's exactly right.
Just clarifying this. Now press on.
Okay.
You've got a lot here, but I could not contain myself to the end because this is just absolutely crazy. And this is the game that these guys play. And then the silly YouTubers look at this and they say, "Oh, these guys got it because they claimed it in their paper." And this is what I say: when you look at the experimental details, it actually argues against it. So when you publish papers, my colleagues who publish papers, the scientists who publish papers, if you don't come clean with this, then what happens when you don't come clean is that the lay person, like the YouTuber, picks this up and is totally misinformed on this because it's actually just the opposite. This is what I'm talking about. This just nails it home. And again, plus there's a ton of user input. Plus this couldn't work because they've got the selective activation. There's all these games that they're playing. So at every move, this says this is not how nature could have done it. Anyway, press on. Go ahead,
Okay.
Dr. Truman.
Okay, so we don't have too much more to say about the other two experiments because we didn't really do anything. The next experiment now has got a gap here where 2 amino acids could add. now it is no longer true that phenylalanine and valine added as had occurred in the previous experiment. So previous experiment, again, it's only valid when there is a single gap but open and nothing else. So now they're finding what I would have expected, and that is the substrate alone now reacts more favorably with the activated amino acids than the amyloid would. Okay, the only exception they found was arginine, but the yield was so small they didn't bother reporting it. So basically what I'm telling you is that the whole trick about making a long template and then they show their substrate and then seeing if that could favorably select which amino acid goes there and whether it's D or L just doesn't work for these larger gaps. So the phenomena they found is, let's say, modest at the best. Okay, and once again,
I mean, what you just explained is it favors it not to go the way
correct.
they wanted,
Precisely
the way they wanted to. The amyloid is not as good as the free substrate alone. So
correct.
So again, it argues against this being the route that this could have taken place in nature. I mean, let's just take the gloves off, Dr. Truman, and just say it for what it is. I mean, this is what it's screaming out to be. I mean, we keep cutting these guys slack, and then they just hang themselves with it.
Yeah, here's kind of my take. What we're looking at here is what chemists are trained to do. You're trained to solve a problem, find a clever way to achieve your goal, and chemists have many tricks to achieve that.
I wonder how many of those chemists. I wonder how many of those clever chemists were around on our early Earth.
That's the point. I mean, that's the point. The point is, this says absolutely nothing about how life could have originated. Quite the opposite. It's just another example of what could not have happened. You know, you're kind of running out of ideas eventually. But I don't want to criticize the cleverness of experiments because, chemists can have
I'll criticize it. I'll criticize it because this clever experiment is actually teaching people, is actually suggesting to people that this is how it happened when it's talking the opposite way. So the poor little YouTuber who thinks that this solves the origin of life problem, he's going ahead and putting out all these YouTubes, and the poor guy is so deceived. That's the problem with it. And then he deceives everybody who listens to his content. I mean, this just condemns it. It condemns it. So I'll go after them. When you put your cleverness into these experiments and it still doesn't work, it works poorly and it could never have shown the fidelity that you needed, never could have shown the coupling that you needed. You set up one condition after another in addition to the concentration and it still doesn't go the way you want it. That's really bad. And then to suggest that this has something to do with origin of life, liar, pants on fire. That's what they are. Go ahead.
Yep.
Sorry about that, Dr. Truman.
No, I understand. And then the experiment number 3 is even less remarkable. Now, when the gap is large enough to accommodate 3 amino acids, they didn't get any amyloids whatsoever. So basically, not much to say here. They did play around a little bit with throwing in 2 amino acids that theoretically, if one of them reacted, it would make it easy for the next 2 to form the beta sheet. So they selected 2 arginine and phenylalanine. They had to increase the concentration very high and got, of all the things that could have reacted, a trace amount. Trace amount meaning no number is, some peak that seemed to indicate that 2 of these 3 some amino acids may have added, and a third, a tiny amount, which had some of the D-phenylalanine mix. Essentially, not worth discussing because anything that is so small here that it would have no relevance for any discussion is not worth spending more time on. So basically, experiment number 1 is the only one worth looking at and only involves one position. in case we run out of time, keep in mind, in experiment number 1, there was one gap position to add one amino acid. That means that you cannot form large amyloid this way because the amyloid can only grow as large as the template. After you fill in the gap here, that's the end of the story. So we're talking about something of extremely, let's say, low plausibility or relevance, honestly.
Yeah, I mean, and plus, even if they got it, even if it did what they wanted to do, these are so ridiculously small that it's laughable. But anyway, yeah, and that's why you're laughing because it is really nonsense. We got to press on here. Go on to the.
Okay.
I mean, how many of these problems do we have to stack up before the amyloid hypothesis bites the dust, just like every other hypothesis that they've put forward? This is really nonsense. But go ahead. Let's look more at these experiments.
Right. The fourth one now is the idea of regioselectivity. That means, is there any way to get the end, in this case, amino group to react instead of the side chain? So now they created a different template. It's made up of 9 amino acids. The numbers are very, very important. We'll come back to this. 8, 9, 7, whatever. These numbers are extremely important. I'll show you why when we do some of the mathematical computer modeling. Okay, so this now has 9, and it has a very, very specific pattern. It's using aspartic acid and valine. Now, for the substrate here, they have chosen something entirely different, and they're using a non-biological amino acid, and here's the structure, orthonine. So, what happens? When they don't provide a whole lot of data. It's more.
Wait a minute. They're not even using one of the canonical amino acids. They've chosen
I'll come back
a loaded one here.
I'll come back to that. Yeah, I'll come back to that.
Okay. This is laughable.
So the theory is, and this makes sense. It's clever. Is that if you form an amyloid here, the side chains are going to be facing each other, and that would protect the side chain. Here's the long side chain, and therefore the argument is, oh, well, that's kind of cool. It's less likely that another amino acid is going to react with the wrong position, the side chain position, because it's embedded in the middle of the amyloid. That's the thinking. And so first they tested what happens if we look at the substrate alone. And they found that, 25% reacted at the end position, which is unsurprising because if you look at the structure here, the orthonine has got many, many more side positions than only the one end position. So, of course, you're going to get more of the side positions reacting in the case of the substrate alone. So 25% of the end is what you would expect. And when they mixed these two things together, waited until the amyloid had formed, and then afterwards added the amino acid to see what happens here. In this case, added valine and phenylaniline. Then, as pretty much expected, only the end group, not only, but more the end group reacted than before meaning the naked substrate alone. So that is whenever you read this catch-all general statement that amyloids have been shown to be regioselective, it's all based on this experiment, nothing else, okay?
And the regioselectivity is what?
You're talking about
75%?
Right. Yeah, it's in the case of phenylaniline, it's 85%. So it is real.
So it is real. But you have 15% going in the wrong direction.
Yeah.
It's dead. It's dead. I mean, if 15% of these are going the wrong way, it's over.
That's correct. But Dr. Tour, the point, the stronger point is a little bit different. That is, of all of the biological amino acids you could have selected, for example, look at the structure of arginine here. Look at
Yes.
arginine here versus orthonine. Okay, you're talking
Yes.
about a difference of one carbon. Why? Why was a non-biological amino acid selected? Well, it's obvious because it didn't work for anything else.
Because it was the only one that would work.
Exactly.
So even in their loaded system, which is totally unnatural,
I know.
it worked so poorly it would have been useless. I mean,
Right.
that's what these guys are doing. This
I
is the
think
game they're playing.
that's the difference between you and me and others. One has to know how to read the papers.
Yeah,
Okay.
the layperson doesn't know how to read the papers. They just read the summary of what these guys are showing, and they said that this has the regioselectivity. It does not. They had to make a fake system. And even in their fake system, it only had 85% regioselectivity. You say, oh, 85% is good. No, in biological systems, that means 15% give you a cross-link, it's over. Once there's a cross-link, it's game over. And so why didn't they use the real one? Because they couldn't. Because it wouldn't work with the real one. So there's another game. Okay, go ahead, press on.
So, summary of these particular experiments. First of all, concerns, to put it diplomatically. The both ends had to be protected, otherwise they would hydrolyze, and of course that means that in water they wouldn't have existed abiotically. It's that simple. The same is true of the substrate, and then the carboxyl end. The amino acids had to be activated. That means the way this works is in the laboratory. You've got to activate these amino acids first of all in a non-protonic solvent quickly before you do your experiments, because the things don't last very long in water, and that's how you do experiments. That means that in water the stuff would never existed. Okay, now here's something to think about. They took substrate and templates already full length. That means they did not allow them to polymerize and to see if they would form at all. And then in this mixture of short and long substrate, short and long mixture of templates, whatnot, see what happens. No, they took two of these peptides of exactly the right length, synthesis in the laboratory, mixed them together, agitated them at 800 RPM, used the right pH, the right temperature, and then waited until they formed the amyloids. Solidification to break them up, make them look nice and pretty. So basically, you've done a lot of lab work here to create the amyloids you want. And then what happens? So you had to use only L-only peptide substrates and templates. Otherwise, you don't get the beta sheets at all. Now, in the right stoichiometry, was, of course, optimized. It turns out you need a little bit more template than substrate to get the highest yields. All this was played around with until they got the best conditions, but roughly 1-to-1 in the same location to allow you to form amyloids in extremely high concentration. I'm going to come back to this in part 2 because I am very, very interested in quantifying credibility and plausibility of some of these things. Okay, and these things can be quantified. I've done this with some of my colleagues. You've got to have very, very precise sequences. Okay, otherwise you don't get these beta sheets, and they've got to be identical. Okay, so you need a very high concentration of identical, very, very rare peptides in, of course, a closed container to keep out contaminants. So basically, that's it. Now, you talked about somebody from the YouTube. I assume you have somebody in mind. It does turn out that there are some videos out there, and one of them will be played as part of this presentation here. It's a 50-second video, and it's referring to some of the work they just described. It is also referring to some other papers, in particular a review article by this Professor Mari, who wrote, and I quote here, that prebiotic amyloids produced a series of biology-like new functions, including self-replication. Self-replication, in case you're wondering, is referring to the experiments I just described. Self-replication means that the amyloid is allegedly forming other amyloids that has a little bit more phenylalanine in it. That's about it. There's another paper I'll talk about that also claims self-replication, but it's a very, very strong word. This is not what we're talking about in biology, about cells self-replicating. Kinetic activities, I'll talk about that paper, why he says that. Information transfer, I'll talk about that. Chiral selective addition, we already saw that. The strong statement here is based on that experiment we just described. And now you have a catch-all conclusion that they have this chiral selective property. And error correcting. So, somebody put out this YouTube presentation, and in this 50-second one paragraph, identified 6 errors.
ATP is another naturally occurring energy source near hydrothermal vents. So we know that amyloids have ATPase activity. They can utilize the energy of ATP to do work. And once you develop this interaction between amyloids and ATP, it's only a matter of time until you start having refinements into the information storing system, the stringing of ATP and different triphosphate nucleotides together into DNA and RNA, the incorporation of lipids, which are again found naturally at hydrothermal vents, and which spontaneously form bilayer membranes, as well as the creation of protein enzymes to do specific functions.
I'm not discussing this next to make somebody look bad, somebody who does put out other, pretty good videos and other topics, but it is representative of a lot of what I call YouTube evolutionary science, okay? It's very, very representative now. Maybe explain a little bit why. And it is dealing, of course, with amyloids. So I'm going to talk about these 6 errors and explain why they're wrong and hopefully avoid other people getting confused.
Okay, let me just give a quick summary here. They had to have the protection, deprotection, the protection activation. This I described in my 14-part series, where you have to have protection to be able to do this. So none of this will happen on an early Earth. There's no selective activation like this. That's number one. Number two, it was totally loaded based on the specific amino acids that they were using. You have the wrong amino acids there, it's not going to work. It didn't give the stereoselectivity. It didn't pick out the right enantiomers. It didn't give the regioselectivity. They had to use a non-canonical, a non-natural system because the natural system didn't work. So they used an unnatural system, and that unnatural system worked only poorly. When 15% would have cross-linked, you have 1% cross-link, and the whole thing changes. If you have a rubber band and the polymers are not cross-linked, the thing is a liquid. And you put 2% cross-link, you get a rubber band. I mean, the cross-links change everything. And in peptides, you get these cross-link, it's game over. They had the concentration. They would have had to have a concentration where they would have had the sky raining pure amino acids to get that kind of concentration in the oceans or in some tidal pool to do this type of thing. They adjusted the pH. The pH has to be just right. You can't have too much acid. You can't have too much base. It has to be just right. I mean, these are Goldilocks conditions. And then the temperature had to be just right. and so they adjust all these things. And the stoichiometry had to be just right, meaning that they couldn't have too much of one versus the other. They got the stoichiometry just right, and it only worked with specific sequences. So you couldn't just have your random amino acids around. This whole thing is loaded. Dr. Truman is very gracious man. He says they're clever. Yes, they're clever, but they're cheating. They are cheaters. These people are cheaters if they are relating this to an origin of life experiment. And the victim of the cheating are these YouTubers who take it and pick up this banner and say, look at this banner. And they run around with this banner as if they've got it. The poor folks are victims of the origin of life community making these claims. That's exactly what I've been saying all along. Okay, sorry for the interruption. I just had to have people understand this.
You're right. Let me add a thought, though. You said that even a small amount of, let's say, side chain reaction, like 15%, would be very, very bad. Here's a thought. What happens as a peptide gets longer and longer and longer, and we said that the short one had 15% side chain reacting. That was 15% of a short one. What happens when now you end up with a peptide of any relevant length? Now you're talking about something, let's say, 300 amino acids long. You have one amino acid at the end that could react. Plus, think about how many side chain positions you now have. So
Yeah,
even if you had only 15% side chain reaction, eventually you end up with a vast majority of side chain reaction, and every side chain position can further react.
Yes, what he's saying is it
branch.
gets worse because
Exactly.
you have so many more side chains hanging off.
Exactly.
As these things get up to reasonable molecular weight so that they're useful for anything, it gets even worse. This is in
Exactly.
the best case. All of these problems I just told you, this is in the best case.
So I'm going to talk about 6 statements and probably add some more afterwards, not to pick on one particular person, but first of all, these are common mistakes. Second, a lot of people hear these statements and repeat them because they sound plausible, but they don't know the chemistry. So what they're doing is believing things that are fundamentally wrong, that every chemist knows wrong. So here's the first one. The 50-second clip begins with a statement.
ATP is another naturally occurring energy source near hydrothermal vents.
Okay, that's just a statement of fact. And it's totally nonsense. First of all, ATP has got 3 phosphate positions, and a glycoside position, that are all exothermic. That means they are unstable. They will hydrolyze in water. They are not going to form. So I wrote a paper, did some calculations with some friends of mine. Basically, here we are, some calculations. The bottom line is at 350 degrees, let's say hydrothermal vent condition, the amount of ATP at equilibrium would be about 2 times 10 to the minus 25 molar times whatever the concentration of adenosine is at equilibrium, which adenosine would be this ribose structure here with the nucleobase at the end of it. That's the base here. So this species here is also highly unstable, okay? So the concentration of this will be very, very, very low, much less than 1, multiplied by this. That gives you the amount of ATP that you expect to form or to find equilibrium in hydrothermal vent. So basically it's showing you here the different equilibrium reactions, the different ways you can split it apart. But if you go all the way back to adenosine here, it's simply not going to go add one phosphate group after the other to form ATP. It's just not true. Here's the idea. Before you can get a DP, the diphosphate, you've got to have the monophosphate, okay? So this is an uphill reaction. So you can see right away what the problem is. You've got reaction after reaction after reaction, all of them, each one of them individually unfavorable. So that's why it's unsurprising that our calculations show that the amount of ATP is just absolutely negligible. It's not, as our YouTube friend claims, readily produced. Okay? like I said, I published the calculations and everything else.
Okay. All right. Just go back before you go on to number 2. Look, if you find ATP near hydrothermal vent today, that means nothing. That probably came from a biological source. I mean, the oceans are full of life. To say that that came spontaneously from a hydrothermal vent on an early Earth before there ever was life is a totally different thing. But now this is where Dr. Truman is an expert. He spent so much of his life, not just as an organic chemist, but as a computational organic chemist that allows you to calculate these sort of mixtures. So he was a chemist for BASF, which is a huge chemical company, and you have to be able to calculate concentrations of things at equilibrium. When he's talking numbers that I think you said 10 to the minus 20 something, did you say?
25, yeah. Minus
10 to the
25
minus
molar.
25. That means it essentially does not exist. It's not
Right.
there. You can't do chemistry at concentrations like this. So that claim of ATP was ridiculous. The victim of that is the YouTuber. He is the victim. And he's going around and carrying a banner and saying this stuff that is utterly nonsense. But he doesn't know. He really doesn't know. I mean, that's why he's, carrying this banner that he's figured this thing out, or the world has figured this out, because of the origin of life researchers themselves. They should have pointed out that any concentration that you would see today is relevant to what would have happened on an early Earth. And even if you had it by a hydrothermal vent, its concentration could not be that much because of its hydrolytic instability. That's the problem. That means they decompose in water, which is what's all around a hydrothermal vent. So anyway, go on to point number 2.
That is correct. Before moving on, then maybe I should mention where I think the misconception, or I should say the deception, is occurring here. There are a couple of papers where people have speculated. They've imagined, well, how might ATP be formed, okay? These are not experiments. This is not that they found ATP from organic sources near a hydrothermal vent, but they're saying, well, gee, let's just think about that. What if somehow there was some kind of a membrane that was able to separate two high and low pH regions with a difference, pH difference of about 4, because that would produce a sufficient electromotive force to enough energy to create the ATP. This is a couple of papers written on this. It would require, of course, a special motor to convert the energy in a manner to actually load the phosphates onto ATP, et cetera, et cetera. There are a couple of papers, and I think, based on some quotes I'm going to give you a few slides later, that this particular YouTuber simply did not understand what was being stated. Another fellow, Jackson, has shredded this whole idea for obvious reasons. It's just pure speculation that simply cannot work. What kind of a mineral barrier are you going to have that's going to constantly allow protons to flow through it with some kind of a motor? And when you look at the dimensions of the membrane, that would have to be stable but thin enough to allow protons to get through. The whole thing is just simply nothing more than a Gedanken experiment. It's just a thought experiment. There's no scientific basis. So I think that is where this YouTuber has got this idea. It's a hypothesis, an idea. There's no science there. So it's not true at all that it's readily available. ATP is readily available in hydrothermal vents. That's simply not true. Now, moving on.
So we know that amyloids have ATPase activity. They can utilize the energy of ATP to do work.
They can utilize energy to do work. Well, this is wrong on so many levels, but just start off basically with the fundamental understanding. If you have a high energy source, let's say fuel. Let's say you dump fuel on a car and you put the car on fire. It doesn't do useful work. It's heavy energy, or in any way catalyzing the degradation of ATP, releasing a little bit of energy, that doesn't create work. In fact, it can do a lot of bad things. So indiscriminate destruction of ATP is not at all what an ATPase does. These are enzymes. They have specific structures. They bring the substrates together in 3 dimensions, the right substrate, the right position at the right time with ATP, and then it allows a specific chemical reaction to occur in a very, very carefully regulated manner. It's not just, randomly diffusing energy. So
this again, let me just underscore this. You get energy when you're part of a biological system in a cell. This thing is so precise in order to capture the energy that's there. Energy by itself doesn't give you anything. You have to be able to apply it. And what a cell does, it allows you to apply this in the right place at the right time with all the other functions around it to be able to capture that energy. So just having ATPase around isn't going to solve anything.
Right. We'll talk a little bit about that also, why the claim that amyloids have this property, where it's coming from, is also kind of a misunderstanding, to be honest. Okay, so now he continues the same paragraph about 12 seconds later.
And once you develop this interaction between amyloids and ATP, it's only a matter of time until you start having refinements into the information storing system.
Like, what? So you've got an amyloid, you got ATP, and suddenly you got a steadily improving information storage system. It's like, scratch your head for half an hour and try to figure out, like, what?
What on earth is he talking about? Help me out here. Help me out here, Dr. Truman.
I think he read, misunderstood some things also written by Dr. Murray, another paper, and here is a statement. And here, let me just read it, quote it. This is what Dr. Murray wrote. Information transfer on the early Earth for about 4 billion years ago occurred according to his particular theory by means of a beta sheet peptide-based prion-like amyloid system. Okay, here comes now, in which environmentally derived information encrypted in the beta sheet zipper structure was transmitted by a templated conformational self-replication mechanism to daughter amyloid entities. Basically, and I think I got some slides, because this is something that has drawn a lot of attention, and I will come back to it. And I wrote a paper with 2 computer science professors, because it is very, very, very disturbing how people that are strict materialists are misusing and misunderstanding the whole concept of information.
Let's not grab all materialists. The most materialists would fall off their chairs to hear this if they were thermodynamicists or informaticians. This is utterly ridiculous and irresponsible of Dr. Morey to be able to write something like this. And again, there are victims of this. The victims are the poor YouTubers. Then they propagate this nonsense to environmentally derived information encrypted. I mean, this is inconceivable. Show me how the environment, how just the environment of a non-biological system is going to make the information that you're going to need and then encrypt that information, and then you then have this information embedded in this that is now transmitted. This is a bunch of nonsense, and you see the outcome of the origin of life researchers, Dr. Morey in this case, infecting the minds of these poor young people that have these YouTube videos where they totally misunderstand what's going on. Anyway, press on.
Right. So I will have a few words about information, because it's something I've spent decades looking at, and it's part of my second career as a computer science specialist, and it's something that I'm currently working on at a German university. So I had to say something. I have to say some things about that. But let's move on about this. Okay, right. Maybe just to complete the thought here. Time. Time is somehow automatically going to refine an information storage system, whatever it's supposed to mean. The whole concept here is that there are physical effects caused by the environment. For example, we know that gas expands when it's being heated and water freezes, when it's cold enough. These are simply physical chemical responses, okay, which has nothing to do with encryption. For example, storing DNA instructions to specify the sequences of proteins. Time doesn't automatically generate goal-oriented information. In fact, just thinking about that for a little bit, the second law of thermodynamics, there is a tendency towards maximum entropy. So let's just say that you had some kind of an information storage system like a hard disk or something. Time is going to corrupt the physical carrier of information, the carrier physically, okay? So here they've confused information as the carrier itself, which I'll go into a little bit. But even the second thought is the carrier of information, the physical medium itself, is subject to entropy. So time is not going to help you. It's your worst enemy.
Time is your worst enemy. This nonsense that time refines things. No, it doesn't refine things. It doesn't. Time works against you. If you get a memory stick
Right.
has flash memory. That's guaranteed for 10 years. Why is it only guaranteed for 10 years? Because those bits of information start getting lost. And any memory system, whatever it is, begins to get lost over time. It doesn't refine this. It gets worse over time. And I mean, this is contra-thermodynamic, what you're saying here.
Exactly. So a way to understand a little bit, and I don't want to belabor it in this particular presentation because it's too long a topic, but think of it this way. The argument that Maury and the YouTuber and many other people are using is analogous to saying that random changes during however silicon chips are produced, okay? The manufacturer, we don't care. However silicon chips are produced would create the variety of computer programs, that the physical effects, that the temperature, the moisture, the catalyst, that these things would create different kinds of programs. And it can't be right because if it were true, one particular program, let's say a database or Excel or a spreadsheet or an AI system, all of these programs are totally, totally different. And so there is no physical way to go from environmental changes into a coded program. That's simply a category error.
Right. And the hydrolysis rate, the racemization rate, every time you have a stereogenic center and that thing racemizes, you're losing information. Hydrolysis, you lose information. These polymers don't last very long in water. So
That's correct.
Information is lost. Anyway, press on.
The stringing of ATP and different triphosphate nucleotides together into DNA and RNA.
The stringing of ATP and different triphosphate nucleotides together into DNA and RNA. Well, it doesn't quite understand how the chemistry really works. The truth is, in cells, you do have DNA and RNA polymerases, all right? These are these monstrously complex enzymes. They indeed do use nucleotide triphosphates, okay, as substrate. That is correct. But that is not how this would work in nature and water, and it's not the way origin of life chemists try to produce RNA and DNA. He's got his chemistry misunderstood. These RNA and DNA backbones are negatively charged, and trying to add some nucleotide triphosphates to negatively charged molecules is simply not going to work. So you do need special enzymes for this to work. In reality, origin of life, chemists, what they do is they activate the monophosphate, okay? So you've got one monophosphate is activated here, and that's what they use to try to synthesize DNA and RNA. So not to belabor the point, simply his chemistry is wrong. Let's just drop it, okay? It's not right.
The incorporation of lipids, which are again found naturally at hydrothermal vents, and which spontaneously form bilayer membranes, as well
Then
as
the incorporation of lipids, which are again found naturally in hydrothermal vents and which spontaneously form bilayer membranes. So now he's trying to go to something that looks like a cell. So you've got a bunch of amyloids and then a bunch of lipids floating around, and now you've got bilayer membranes. It's like, what are you talking about? Like, what is going on? What lipids are being incorporated into what? What are you talking about?
What he's suggesting is that you can get these lipids coming out of hydrothermal vents, and you can. Lipids can come out of hydrothermal vents. You can get reactions to occur, and they need not be of biological origin. What's going to come out today would mostly be of biological origin, but they need not. And you can tell by looking at you can tell by looking at the nuclear content, at the radioactive content, or the concentration of different isotopes that are there. So you can tell if they're of biological origin or of non-biological origin. You're not going to get ATP, which you were saying before.
Right.
That's not going to happen. But you may get some lipids. Now you need really for a really stable vesicle, you need diacyl lipids, not just any lipid. But with any lipid, you get something that is very weak. It does not allow you to form the proton gradient across its shell. It does not have proteins that are going through there. So what you would get, if you get a lipid, it would not be a diacyl lipid, so it would be very unstable. Plus, it would be extremely leaky, so you're not having the proton gradient that you would need. So in other words, it is not a protocell. These things that are coming out of a hydrothermal vent do not give you the protocells that you'd like to be able to call a protocell. So he's got that wrong as well.
Right. So you've pretty much summarized very well the last 2 points here. The first is these lipids would have to have a charged ends. They have to have they're very special structures, and they have to be at a very high concentration. We talk about the critical aggregation concentration or the critical micelle concentration in order to form micelles or
right. They don't come out of the hydrothermal vents at the concentrations that you would need, the critical concentration that you need to form a micelle. And plus, what you have to do is you have to start applying shear to these, a shear gradient to these, to be able to force these together to get them to really form. And you just need much higher concentrations of them. And you also you can't have a diversity of different lengths of lipids, or else it's again going to be unstable. So whatever you might form wouldn't even be very stable, and it wouldn't give you the proton gradient that you need to be the structure around a cell. So you got to distinguish these things, and the poor kid just doesn't know what he's talking about.
Exactly. Now about the ATP, what would happen here is if for whatever speculative reason, you would form some kind of a bilayer membrane around some kind of an amyloid. What would happen then is, as you already indicated, if there's a misbalance in the proton concentration, it eventually equilibrates very, very quickly, and now there would be no electromotive force to permit the creation of ATP anymore. So, and there's simply there's no logic to the argument here.
As well as the creation of protein enzymes to do specific functions.
So, somehow you've jumped from amyloids to now you've solved a problem origin of life chemists have been unable to solve, and that is, where do protein enzymes come from?
So he just pulled that out of thin air, right out of his hat.
Exactly. Look,
Yeah.
the typical enzymes are on the median order of about 300 amino acids. Let's just say the smallest one's 100. The synthetic amyloids that we've been talking about are all less than 100 amino acids. And the two simply have
What
no
they were
connection.
showing in their work were things like that were like 8-mers.
Right.
Yeah.
So, and there's simply no logical connection. You've got very, very specific sequences forming amyloids that cannot grow or in any way change. And how do you go now to something entirely different? These monstrously long, complex enzymes. There's no logic there.
And properly folded. If he is going to grab a protein enzyme out of his hat like that, why don't we just grab a cell? Why don't we just grab a human being? And just say, hey, there's life. There's a human being. What else have you got in your hat there?
It's more that would be more plausible because time is a big enemy. If you're going to argue a miraculous course of events, at least everything would be in place quickly and it would at least work. But if you're going to argue that over long periods of time these things arose, that's even less plausible.
Yeah.
Okay, so let me quickly wrap up just to make sure that this wasn't kind of a bad 50-second period where, a little dementia. Few more massive mistakes, then we'll move on to the next topic.
Let me break this down as simply as possible. All of the building blocks that go into the formation of amyloids have all been shown to be naturally occurring.
Now, the claim that all the building blocks that go into formation of amyloids have been shown to be naturally occurring. Okay, let's stop with that. Unfortunately, 99.9% of all YouTubers don't read technical papers, but these kinds of statements are easy to remember. Oh, everything is naturally occurring and to get amyloids. That's easy to remember, and they're simply crazy. Look, they started off with homochiral peptides that were capped on both ends, and the amino acids were artificially activated. There's nothing naturally occurring here at all. And I'm going to come back to this again, before we quit today. The peptides used constantly rely on amino acids that evolutionists believe did not exist prebiotically or in trace amounts. They rely on this. So you're not going to have these kinds of peptides at all. So it is not true that these have been shown to be naturally occurring.
So you have these amyloids being formed with essentially random sequences.
He says amyloids being formed with essentially random sequences. There's nothing random about the sequences these very clever chemists engineered to work. L-only or amyloids using a template and a substrate strand very, very cleverly align precisely with the right amino acids, the right position to form an amyloid. These are not random sequences. Okay, nothing could be further from the truth.
Yes, and this statement that you have at the bottom.
That's where he got it from. That comes.
That's probably where he got it from. Yeah,
he got it from Maury.
Uncoded short peptides, and then you get this error-correcting replicative cycles generate amyloids that represent the first coded peptide polymers. You might as well pull that statement right out of the hat as well. I mean, where this researcher got this thing? Is this Murray again?
Right. This is Maury. I see. Look,
boy, this guy's a piece of work.
Okay, look. Let me. We've got to be objective fair about this. Murray is not saying this is true. He's saying one can visualize, one can imagine. This is a Duncan experiment. It's a thought experiment. He is not saying that this happened. But unfortunately,
he wrote it, and then the poor YouTuber takes it as gospel.
Exactly.
Yeah, this is the danger with you guys. You guys are going to you say these things, look at what it catalyzes. This is the problems. Murray is he is the guilty one, I think. He is the guilty one for having even written it. that you can imagine this? How can you imagine this? You can imagine all sorts of things. You can imagine, people just dropping out of the sky. I mean, you can imagine it. I mean, what does this mean?
Right. And then the last one, I mean, I don't want to belabor the point. I mean, it's a very short video, and believe me, there's a lot worse stuff out there.
And the great thing about hydrothermal vents is that they make their own proton gradients. In our bodies today, the reason why we need to breathe oxygen is to establish proton gradients. But back at the prebiotic Earth, proton gradients were formed naturally at hydrothermal vents, which are key and integral in forming ATP in the first place.
He presents again the pH gradient as self-established, as obvious, as established fact. Okay, and again, it's nothing more than another researcher, not Maury, it's another researcher saying, well, we can kind of imagine. That's basically it. Okay.
I see. So he takes people's imagination and he makes it reality. Okay, I got it. Yeah, and even a pH gradient in a hydrothermal vent, how's that going to do it for you? I mean, this is crazy. It's just really crazy. Anyway.
Right. So moving on to another paper. I'll try to go through this quickly, but the slides will be helpful if a person is interested in the topic. And I've written a couple papers on this. Some more will be coming out because it is important. In this, Maury is making a lot of claims about information, other things. He really wants to claim amyloids can store information, process information. He uses statements like they're involved in information transfer, error correction, and things like that. But the fundamental thing is this notion that information is derived from the environment. And this is
ridiculous.
Serious.
So Murray says that information is derived from the environment. Right. Boy, I'd love for him to show that. And the information that we saw on the last part, actually the information was randomized. He couldn't get the right isomers to go in there, and he had 15% of the wrong things, and then he had the wrong stereochemistry as well. His experiments showed randomization. They didn't show any fidelity, and where information just comes randomly from the environment. Boy, I'd love for him to be able to justify that.
I'm going to try to not spend a lot of time on it, explain a little bit the thinking, because a lot of people are saying this, and try to explain it a little bit. It's a play on words. It's a misunderstanding of the concept, the fundamental concept of information. And trust me, this is something I know about. In cells, there are dozens of codes. The genetic code is only one. There's also a gene regulation code when the mRNA is expressed, the NN rule, the histone code, various sugar codes, the microRNA code, splicing code, forgot the comma here.
Every polymer in nature is coded. Every polymer. Whether you're talking about saccharides, nucleotides, or polypeptides, every one of those is coded. As soon as the individual molecules are the letters, but as soon as you have a polymer in nature, it is coded for that. It has a code.
Mm-hmm. Right. Now, the different kinds of codes in cells, and I've also written some papers on this, and this is what I'm currently working on at a university here in Germany with a bunch of other people. The different kinds of cellular programs, okay? There are many, many kinds of different programs, different codes, okay? This whole thing is closely related to our discussion about the origin of information. I want to just share a little bit because I think for those who are interested, this can be helpful. It is true that physical chemical changes have an effect on molecules, okay? So people colloquially in everyday speech, they often will say stuff like, well, we collected information about the stars from their light, or we collect information about the environment. This is not what we mean when we talk about information and programs and cells and computer science, okay? Information was provided or collected. That is not information, that's data. Data was collected, and then it was interpreted. It can be interpreted, it can be stored in some symbolic manner like text or in a computer, but information is not provided or generated by the environment. For example, we have smoke shapes. Smoke is affected by air currents and temperatures, stuff like that, okay? That is correct. But if you want to understand the meaning of American Indian messages from the smoke signals, okay? You will never understand this, and this is not deriving from the physical things that can change the shape of smoke. They're two different things. One is the symbolic use of the size and shape of smoke to mean whatever you want it to mean. The enemy is coming, the buffaloes are nearby. This is the meaning. That is the information. But when you talk about information about the environment, what people like Moore are talking about, they're talking about the environment affecting something physically that has nothing to do with coding, with programming, with information.
Well, there's this Shannon information that can come from a random arrangement, but what we need is something that Leslie Orgel used the term specified information. People will say that specified information is a term that somehow was pulled out of thin air by creationists. It was not. It was that specified information was used by Leslie Orgel, and actually, I think even others before him used that term. And that sort of specified information, you're not going to just get randomly. It's just not going to happen.
So I'll try to wrap this up fairly quickly because I don't want to get sidetracked on something that is very close to my heart, but it's not the central point of today's discussion. If you want to know about information and the theory and all the properties, a very good name is Professor Gitt, who has written a lot about this. I would recommend his writings. There are some fundamental ideas that one must not forget. First of all, codes programs must be independent of the carrying medium. You mentioned a memory stick, for example. The content of the memory stick can be photos, it can be an AI system, it can be anything. But that is not defined by or created by the properties of magnets or anything else. They're two different things. So whenever you've got codes, by definition, the codes must be reassignable symbols. They're not things that are changed by solvents or temperature or things like that. So this is the whole fundamental essence of a code. It must be able to represent something. One last slide, just to kind of nail the idea. Let's look at what's happening in a cell. The codons of DNA, which specify the proton sequences, they don't interact at all in any manner with an amino acid anywhere. How does this work? The DNA is used as a template to create a copy called the messenger RNA. So here you've got the messenger RNA, right? And here you've got these triplets called codons. So a pattern of 3 nucleotides defines a codon. Taken together, they have a meaning. Now, here you have what's called an adapter molecule, a transfer RNA, right? At one end, you've got the anticodon pattern, and at the other end, you've got the appropriate amino acid attached. That means that these transfer amino acids, they're basically seen if this end here matches is the counterpart of this codon. If it is, then it's dragged into and attached to the ribosome, and then the end here, the amino acid, is then attached to the growing protein. So at no time is either the messenger RNA, and much less a DNA, ever physically attached to the amino acid. So there's no physical connection there. Fortunately, the system could never work. This is a code. That means you could have had many different codes to solve the genetic system. In fact, you could have, I wrote an algorithm, 10 to the 84th different genetic codes that would have worked. That means what pattern of codon do you want to have represent what amino acid? You could have done it any way you wanted. You could have replaced this particular anticodon with something else, and that would then automatically bring in a different amino acid, or vice versa. You could have left the transRNA the way it is, but have the enzymes put on a different amino acid here. So there is no connection between physically DNA or RNA sequences and how protein sequences form. It's a real code. It's not a physically specified mechanism. Never was.
Just not to get confused here, I just want to be sure. So the DNA does prescribe via messenger RNA the code for the peptides, but the DNA had to have had some inherent code, and then it's transferred to messenger RNA, and that's transferred to the protein synthesis, correct?
The code is a priori. The code is the meaning. Yes. The code is
the physical material. The physical material is like the memory stick. You buy a memory stick. There's no information on it. It's void of information. It's not until you apply some information to write something to it. Now it has a code on it. Yeah. And, this is an analogy. So memory sticks do come from the factory with a little bit of internal code on them, but the factory has put that on there. And then there's nothing in there inherent in that flash memory transistor, which is a typical transistor with a deep trench capacitor.
Correct. The exact same bits on a memory stick could be used to mean different things. It depends on what the program is or the code is you choose to use. And that's maybe just one last thought here. The same is true here also. DNA itself is used for many purposes. It has many codes. Some codes are used to specify the sequence of proteins. Other codes on the same DNA are used for other purposes, like to indicate where to start transcribing the messenger RNA, or to specify when to express it, when to block it. These are regulatory codes, all on DNA. So it's not the DNA that specifies the outcome. It is a code that was first planned and it was implemented on top of that medium. And then you put the equipment together to make it work.
Yeah.
Okay. Be careful that I don't get sidetracked because, like I said, I love these topics, but it's not the main topic today. So maybe just a few more slides, and then we'll wrap up part one. Part two will be shorter and a lot more fun. Another study here. Amyloids are supposed to have esterase catalytic properties. And this goes back to one of the claims you saw before. The amyloids have these kinds of ATPase properties, et cetera. It's all coming from this study. So there are an important class of enzymes that rely on 3 histidine amino acids to bind a zinc cation. And the different enzymes for different functions, but it's based on that motif. In this study, 14 heptapeptides were synthesized with a specific structure, which they knew would bind to zinc And they were designed then to form large amyloids. And once again, of course, as always, the things were end capped or nothing would work. So, and what was the reaction that they were looking at? It's a standard lab reaction that's used to compare the catalytic ability of enzymes. It's the hydrolysis of petronitrile phenyl acetate here, catalyzed and it formed, and then it loses water. It loses the acetate, okay? So there's a standard lab test that measures how fast that occurs, and that was used here. And here's the data of these studies. So you've got a bunch of peptides here, 14 in fact. So let's jump into what they got. They did find, after a lot of experimentation, one variant that had, a reasonable catalytic effect, far, far less than real enzymes, but still, it wasn't measurable. About half the things didn't react at all. But let's go to the bottom line.
So what are they trying to say?
They're trying to say,
so what?
Right, exactly. They're trying to say that these amyloids are able to catalyze this particular reaction to some extent, and therefore, because it could do this, then it's analogous to other kinds of similar biochemical processes.
This is
it's
so
nonsense, but it's a lot of fun, okay?
I mean, just a proton or hydroxide will knock that thing off so fast. I mean, you have a nitro group there. That thing is so electron withdrawing. It's just...
Yes, you know more than most people because you immediately saw through it. Virtually any acid base will do it. You're right. But still, they're having some fun. Let them have a little fun. Let's talk about what I did particularly find good about this particular experiment. Let's start off with the fact that they used zinc concentration about 1 million times higher than found in oceans. They had to do this, or otherwise virtually nothing happened. And
so it had to be associated with zinc to do this.
Right. And a very, very high concentration. And guess what? Zinc alone will catalyze the reaction. But of course,
that's what I was going to say. Zinc is a Lewis acid. Zinc will do the same thing without the polypeptide being there, without that oligopeptide.
Exactly.
Zinc by itself would do. Did they compare it to zinc alone?
Nope.
No?
No.
Oh, that is so bad. Is this Morey again, or is this somebody else?
This is Rufo and his colleagues.
So he didn't. He's adding zinc to the oligopeptide, and he's looking at the catalytic activity.
Right.
But he didn't just try it with the zinc alone?
No, but they did do some where they looked at the effect of various amounts of zinc, but not zinc alone, various amounts of zinc. And of course, when you decrease, it's got to be like 1 to 1 or 1 to 2. It's got to be an insanely high proportion of zinc. Then the catalytic effect goes down very, very rapidly with less zinc.
Zinc alone, zinc in water will hydrolyze esters. It's a Lewis acid. So
exactly.
this is silly. And if they didn't do that, I find it hard to believe that they didn't use that to control. But if they didn't, shame on them, then all of these results mean nothing.
But they did. But I got some good news for you.
What?
These guys did do one good control. And it's like you're not gonna believe this. You actually find some people that let's just say did the right thing. They took their very best catalyst here, this particular amyloid with both ends blocked, and guess what?
Is it a thremer? I'm trying to. How long is this quote-unquote amyloid?
7 or 8. 1, 2, 3, 4, 5, 7.
7. Okay.
But guess what? These guys actually decided to do an experiment without the ends being blocked. Can you believe it?
Without the ends being blocked.
Yeah. It's like it's got to be the only experiment in the world where they actually did the obvious experiment.
Yes.
And of course, they got nothing.
They got nothing. So in other words, in every case you had the acetate there, and in which would not be the normal system, and then in the
Right.
one case they didn't have the acetate
Right. So without the blocking group, nothing happens.
without the acetyl. Okay, so and then they use the quote unquote natural system, and it doesn't work.
Right.
So this is exactly what I'm telling you. This is why I say their own work, Rufo's own work, cries out. This is not how it happens. Hydrolyzing an ester is so easy. Just zinc. Just zinc alone will do it. Just a little drop of acid will do it. Just a little drop of base will do it. Just water will do it. I mean, the hydrolysis of an ester, and especially that ester, the paranitro acetate, the paranitro-acetate is highly,
withdrawing group, yeah.
That nitro electron withdrawing group makes water want to attack that very quickly, or hydroxide attack that very quickly. So they took a loaded system. They did absolutely nothing interesting. Their non-natural system will work, and they didn't even do the control with zinc alone. So this paper is garbage. And if the poor YouTuber used this as an example, the poor guy, I mean, it's just sad. It's just sad what they've done to that young man's mind.
Dr. Tu, don't forget, these guys need papers, okay? Don't be too harsh. I worked in industry. I don't have to publish. I got rich just doing honest work.
You
These
think
guys
I'm
have to
being too
publish.
harsh. You know, last week I interviewed Tangelo Grosso, and he says that I'm not harsh enough. And you say I'm being too harsh. Anyway, go on.
Yeah. So a quick summary then. So again, I'm trying to be diplomatic and sort of seeing the positive side of things without being, as the Germans say, blue-eyed, which means too naive. For example, replacing just one histidine eliminates virtually all the catalytic ability. It can't eliminate the zinc, of course. That's still there. Histidine is a rare amino acid, and it's not considered to possibly been available abiotically. For example, you don't find it in meteorites, you don't find it in Urey-Miller experiments, etc. So the whole thing is reliant on a precise structure involving several histidines, which would not have existed at all abiotically. Mm-hmm. Okay, and to create these things, of course, they were synthesized in the laboratory, which meant the side chains had to be blocked. Otherwise, you're not going to get anything.
You
blah,
could
blah, blah.
never make what they're trying to make just through their templated synthesis, because it would be a mess. So they had to make everything synthetically. They had to make everything non-abiogenesis related.
That's correct.
So prebiotic synthesis related, they had to make everything this way. The whole thing was loaded.
Correct. Now, there is an elephant in the room, and I'll come back to that in part 2, which I promise will be shorter and a lot more fun presentation. The elephant in the room here is, how do you get a high concentration of identical peptides that rely on non-existing or very low concentration amino acids? If, for example, we said that this peptide is using 7 amino acids. So theoretically, the variety would be 20 amino acids to the 7th power, about 1 billion different sequences of heptapeptides, theoretically, of which, of course, about 1% would be, of course, homochiral. Now, unfortunately, things aren't quite that good. It's far, far worse. You've got to form all of these peptides, avoiding glycine from sneaking in, even though it's by far the amino acid in greatest concentration. So what you really have to do here is look at the probability of getting the right peptide sequence. So just to give you a quick calculation, when you look at the relative proportion of amino acids that are thought may have been present abiotically, look at those proportions, and that gives you then the correct probability of getting a single one of those sequences, right? So these are the kinds of probabilities, and now you've got to have these kinds of concentrations, right? All, of course, homochiral. So I want to come back to this because it's very, very entertaining and very enlightening to do some of the quantitative work. One or two slides, I think, and then we're done with part one. Just for completeness, just one slide here because it's not worth spending time on. And a study by Nanda et al. And this is the whole idea that's often claimed that systems, networks of reacting amyloids can self-correct, can do wonderful things. Specifically, here's a quote, equilibrating networks of peptides can correct errors by decreasing the proportion of side chain products and the amino acid configuration. Okay, what's going on here? Here you've got one peptide, and you've got another one. These two are designed to form a bed of sheets. So, they combine if done in the laboratory properly, and they form amyloids. So far, so good. Now, here's the issue. This amino group here can attach two positions. The carboxyl end here, which of course is activated, or the side chain here, which is not activated, two positions. And because this is the chiral center, that means you have four possible reaction outcomes. You could have the D or L reacting either at this position or here, and that's why you've got the four products. Here's what you want. You want the two L amino acid positions to react at the end position of the amino acid and not the side chain. So you want to avoid all these other things, okay? So this is the experiment they were doing, and they are making a claim, which is often repeated and, of course, embellished, that these things can correct errors, just like DNA in cells. There are enzymes that can correct errors. Well, they're saying this system can also correct errors abiotically, and they have a way to avoid the D-amino acids, which would be, of course, very interesting, and to avoid the side chains. All of that is based on this experiment. So what did they do? They created, like I said, these long peptides, obviously with a protecting group and an activating group to make the reaction occur at all. And then they did some kinetic analysis and then put together a MATLAB model, a kinetic model with 40 equations. So this whole thing is based on a model. And that predicted that if you had that under certain concentrations, certain temperatures, that it would be possible to get more of the one you want. That is what was published, and that is what was widely quoted and celebrated very, very widely. But here's what the reality shows if you bother looking at the paper and the data a little bit more carefully. First of all, this effect of getting only this product you want, meaning the N group reacted and only the L enantiomer, that only occurred if the reaction occurred at exactly 250 micrometers. Otherwise, you end up getting the wrong product, right? Molar. And only if you let it run for 0 to 2.5 minutes. You let it run a little bit longer, and you end up getting all the wrong products. They didn't say that, of course.
Well, they like time. They like time. So why would it stop after 2 minutes? I mean, they love their time.
I am giving away my deep, dark secret on what to look for in any.
I mean, they're just telling us the kinetic product. They're not giving us the thermodynamic product. And the vast majority of things in Origin of Life are going to give you the thermodynamic product.
Right. And again, you can get kinetic effects by playing around with the right concentrations at a given period of time. But this is not what you're going to get after a little bit more minutes. And of course, they did not do the experiment, clearly. It was only a mathematical model. So time is something you always want to look at in any origin of life paper. Why was the experiment stopped at 47 seconds? Why did it go on for 3.5 days? Always look at that, because you will inevitably find that if you let it go a few more seconds, a few more hours, depending on the experiment, you end up degrading the material and not getting what you want. So time, always look at time when evaluating these papers. So, 2 more quick slides. So basically, all the experiments that I've talked about so far and all the ones that you will find about the amyloids, they have certain characteristics. You want the peptides to be as short as possible. They have to be expertly designed. Otherwise, you don't form amyloids at all, especially if they're short. Man, it takes years of experimentation and chemical intuition to come up with these short
So this would never
peptides.
happen in nature. This is
Right.
totally loaded. Here's another loaded
It's
thing to
loaded.
do.
So you look at all these papers here. Look what they were using stuff like phenylalanine, glutamine, proline, et cetera, et cetera. These are all things that are not found in the meteorites. They are not found in the Urey-Miller experiments. And more importantly, I'll talk about some experiments. They're showing that the most plausible energy sources produce almost exclusively glycine, nothing else. So these amino acids would not have been available to form the correct peptides. Okay? And very, very importantly, you have to form the amyloid first. And only after the amyloids are stably formed, then do you add the activated amino acid. And this is not at all what would have happened naturally. First, the peptides have to form. They start off as a monomer and a dimer, et cetera, et cetera. And during this interval, if the amino acids were activated and could react, they would do so. So that means they would have long since have reacted with the naked substrate and not waited around until these monstrously high amyloid concentrations had arisen, which is impossible, to then and only then react. So that's basically it. The summary of part 1, which is by far the hardest part of the talk, is no life-relevant chemical effects have been demonstrated. There's no concept about a variety of kinds of amyloids interacting together as a system or ecology. This is supposed to be the Amyloid World Hypothesis, not the amyloid crystal, a single amyloid. It's supposed to be a variety of amyloids somehow interacted together, each one based on totally incompatible amino acids. So there's no concept, and there's no concept for a path going from an amyloid world into, let's say, an RNA world, which is frankly even worse, and then from there to a DNA world. So it's not at all clear what people think they've accomplished. I'm afraid, is the bad news for part 1.
Well, another one bites the dust. This amyloid hypothesis is a bunch of nonsense. It really is nonsense. And YouTubers that propagate this, I don't blame them. They are victims. They are victims of this guy like Murray and his ilk that are putting these reactions out there and somehow suggesting that this has something to do with the origin of life. And then they make these statements that you can imagine such and such, and then the YouTubers see that statement and they take it and they run with it as if it's the truth, rather than this is just the guy's imagination. I can imagine lots of things. I really can imagine people falling from the sky.
Very good.
I can imagine that. It doesn't mean it has any basis. So, there's just one problem after another in this. Everything was loaded. This is why I say life could not have happened this way. This shows you that these things don't just happen by themselves. They don't just happen spontaneously. Life could not have happened this way. Well, thank you, Dr. Truman, for this overview, and we'll look for part 2 at another time. And thank you for joining us today.
My pleasure.
Thank you for joining me today. If you could give us a like, share, or podcast review, we would appreciate it. If you have any questions, you could send them to ask@jesusandscience.org, and we'll try to answer some of those questions in an upcoming video. And if you do not believe in the physical resurrection of Jesus Christ, and you want to hear about why I believe, send me an email to tour@drjamestour.org, and we'll get together by Zoom, and I'll share with you why I embrace the resurrection of Jesus Christ.